Hello, and welcome to today's webinar, organized by The Pathologist and Akoya Biosciences. I'm Michael Schubert, editor of The Pathologist, and I'm pleased to be moderating today's online event, in which we'll discuss the association of COVID-19 inflammation with activation of the C5a C5aR1 axis. Please allow me to introduce today's speakers, Dr. Oliver Braubach and Dr. Romain Remark. Oliver is Senior Application Scientist at Akoya Biosciences. After doctoral studies at Dalhousie University and postdoctoral appointments at Yale and the Korea Institute of Science and Technology, he researched optical imaging at Cedars-Sinai. Now he spends his time developing applications for the Codex imaging system workflow. Romain is a team leader in the Translational Immunology and Technologies Department at Innate Pharma. Prior to his current role, he obtained a PhD in immunology by studying the immune environment in cancer. Then, as a postdoctoral fellow, he developed a new multiplexed IHC method and studied the role of anti-GM-CSF antibodies in Crohn's disease before joining Innate Pharma in 2016. During the webinar, Romain will discuss the Explore COVID-19 study methodology, the C5a C5aR1 axis, and its role in acute respiratory distress syndrome, and how C5a C5aR1 axis blockade may play a role in protecting COVID-19 patients from lung inflammation and endothelialitis. The speakers will be on hand to answer any questions that you may have at the end of the presentation, but please remember that you can submit questions at any time throughout the webinar. So without further ado, let me get today's webinar underway by passing you on to our first presenter. Oliver, over to you.
Thank you, Michael, for this lovely introduction, and thank you all for attending. And of course, thank you to Romain for presenting to us today. I would like to take the first 12 or so minutes to introduce spatial biology, or a concept that we have at Akoya Biosciences, and how we think this fits into the current global health crisis, which needs no further introduction than this image. So COVID-19, as I've made these slides, has affected 35 million people worldwide. Not much more needs to be said about that, except that we as scientists today understand that it's absolutely paramount to understand the immune response to COVID-19 if we are to develop effective therapeutic interventions to this pathogen. What's happened since we first learned about this virus is that the pathologists have become very active in characterizing tissue responses to COVID-19. What you can see on the left-hand side is a cohort study from patients in New York and Italy, in which they have detected COVID, which you can see here with monochromatic immunohistochemistry in pink, in the trachea, top image, and in the lung, in the bottom image. The lung tissue that you can see here is very much damaged. There's a lot of evidence of hyperplasia. You can see in pink, COVID infection, a lot of dead cells. We know that this virus does a lot of damage to the tissues, but we don't exactly know how. On the other side of the spectrum, you have molecular genetics, like single-cell sequencing. You can see two images here on the right-hand side. On the top right, you can see individual immune cell populations that were identified via single-cell sequencing, color-coded, where individual dots represent individual cells. On the other image, in blue and red, you can see these immune cell populations in COVID patients, shaded red or orange, or in healthy patients, shaded blue and light blue. The immune responses are very different, evidently. But exactly how this relates to the tissue damage that we see in classical histopathological analysis, we don't really know. This is where I would like to introduce Akoya Biosciences and Akoya Spatial Biology. Our spatial biology, in a sense, is high-plex to ultra-high-plex immunohistochemistry. And it is a very powerful tool for analyzing the immunity with high dimensionality and single-cell spatial resolution. So what you can see here is a lung tissue from a cancer patient, actually, labeled with 6 antibodies. I switch over to the next slide. It's the same lung, labeled with 5 more antibodies. With this technology, called Codex, you can resolve up to 40 biomarkers in a single tissue. Moving on, switched again, same lung tissue, another 6 antibodies. Everything that we do is at single-cell spatial resolution. You know where your cells are in your tissue. You know what every cell labels with. These experiments can be done with ready-to-use reagents and validated antibodies. You've noticed I switched over once again, 5 more antibodies, same tissue. This is really rich information, really rich anatomical and biomarker information. Hence, the data sets are complex. And it wouldn't be easy to analyze them, but we also provide you with built-in image processing and analysis tools. So as I toggle on here, I arrive at our portfolio of products. What I've just shown you is a data set from our Codex technology. You can see that on the left-hand side of this spectrum. Codex is ultra-high-plex immunohistochemistry. You can label a tissue with up to 40 antibodies. Now, I will not go into the details for this technology, but I will focus on our Phenomics technology, which is the technology also that Romain used, in his experiments, in which he's investigating the tissue responses or the patient responses to COVID. So it is instructive to review this technology a little bit. So Phenomics is for medium plexing, about 8 markers. It provides you with single cell resolution. It's whole slide multispectral imaging, and it is very high throughput. And I'm just going to review some of these key points before passing the torch here. So the Phenomics chemistry, which we call Opal Chemistry, is a tyramine signal-based amplification. It gives you a much higher sensitivity of detection than conventional immunohistochemistry. So this left-hand image here, you can see what conventional immunohistochemistry looks like. You'll have an antigen, then you have a primary antibody here labeled in lavender color, and a secondary antibody that has a fluorophore that's labeled green. On the right-hand side, you can see TSA-based amplification. Once again, there's a primary antibody, but your secondary antibody is then labeled with HRP. HRP reacts with our Opal reagents, and it causes these Opal reagents to covalently deposit or label the epitope, the antigen in question. And it does so at a much higher degree than a conventional 1,2 immunohistochemistry would. Opal survives stripping, which means that you can do several rounds of secondary antibody label in sequence, and thereby multiplex your tissue with 6, 7, or 8 antibodies. Stronger labeling means less exposure time. It's gentler on the tissue, and you can multiplex antibodies that were raised in the same host species. Lastly, it is nice to note that this technique is compatible with RNA detection, such as single molecule FISH or RNAscope. The Phenomics workflow is definitely worth mentioning here. This is very important for analysis of pathological samples because data need to be robust. And what you can see here are 2 sets of data. So let's look at the top hand row here. You can see the same tissue on the bottom hand row again. And if we look at just a yellow image on the right-hand side here that says CD8 Opal 480 at the top, and then compare that 2 images down, it's the same image again. However, these are acquired and processed via different workflows. So what we do with the Phenomics workflow is we capture the full Opal fluorescence emission spectra. So that gives us information about the full spectra for each one of the 6 fluorophores plus DAPI that was imaged here. And what we do is we build multiplexed or emission spectra libraries and background libraries. These libraries are then compared to what is expected or what should happen in your, say, Opal 480 channel, and are then subtracted. And what results out of this is an image that is not only much cleaner, but has a much better signal-to-noise ratio. So let's look again at the top here at the Opal 480 image. Look at these 2 arrows. They're pointing at some vascular structure or some connective tissue maybe. Now move 2 images down, straight line to CD8 Opal 480 on the bottom. You can see that this image has been cleaned up substantially. The confidence is much higher. The signal-to-noise is much higher. This is an excellent tool for unambiguous and robust identification of anatomical features. And that is what the Phenomics technology can do. Moving on, it's very important, and especially if you think about clinical studies or clinical pathology, to assure that these technologies are also throughput, that they have good throughput. You can see on the top hand top left here, you can see our Vector Polaris imaging unit. This allows you to do 7-color Opal Polaris reagent kits. So these are ready kits to analyze immune cells in cancer, for example, but it can do up to 9 colors. The system is incredibly fast, 6 slides per hour for 20x resolution magnification. And I should mention it's whole slide imaging, so you're actually imaging the entire slide. You can load this up with up to 80 slides, so this is high throughput. Essentially, in 12-hour sessions, you can image an 80 full slides. Now, there's a lot of data. It's fully enclosed and automated, so it is a perfect tool for clinical and pathological investigations. And you can also do ROI imaging, high resolution 40x, and it can be used for RNA FISH. Now, all of these data are nice, but it would be difficult to appreciate if you didn't have the right data analysis tools. So what you can see now on the right-hand side are data come out of Electra Polaris. These are unadulterated data. That means that they contain all of their background fluorescence and all of the unwanted artifacts. The system isolates background fluorescence, which you can now see in black and white on the right-hand side, and removes it and gives it a multispectral image, the cleanup image. And you can see that the contrast, the signal to noise, is just remarkably much better. This, of course, lends itself much more to digital image analysis and to understanding what is going on in your tissue. We have built-in tools for regionalization, regional identification, so you can compare stroma versus cancer tissues, or vasculature versus cell populations. Individual cells are identified via multi-parameter segmentation. This is a very nice tool. It segments both nuclei and membranes at the same time, and thereby allows you to pick out every single cell and phenotype them. So you can see here, cells are now denoted with little dots in red and blue and yellow and cyan. Those are different types of cells that were first segmented, then phenotyped using machine-based learning. Lastly, with all of these data in hand, it is easy to quantify cells. It's easy to find them. It's easy to know how many there are and how they're different in condition A versus B. So that being said, this is a quick overview of the technology that we call Phenomics, and that's also the technology that is featured in Romain's work. And to conclude, I just want to show you this image. This, again, is a piece of lung tissue. This is a cancer patient, and you can see in cyan are epithelium and or cancer cells. And in yellow, orange, red, magenta, and green, you can see different immune cells or checkpoint markers. The image is clean. This is really revealing what's going on here. But if you think about, this is a whole slide scan. So as I move on here now, I'm zooming out from the same tissue, and I'm putting this into a perspective. And the amount of information that is contained in this single whole slide session is immense. There's so much to learn. There's so much to see at excellent resolution. So I think this is a very, very nice example of what we at Aquarius call spatial biology. It has high dimensionality. It has single cell resolution. It's robust, it's automated, and it's high throughput. And in our eyes, this is a very, very good tool for comprehensive biological discovery that is needed today to understand what is going on when people are infected with COVID-19, for us to build that knowledge base that we need to understand immune responses to this pathogen, to this virus, and to develop effective therapies. So with that, I would like to now introduce Romain once again. He will speak to us about his work that's actually on COVID-19 and immune responses to it. And I'm very much looking forward to what he has to say. So thank you so much for presenting this work to us. And without further ado, here you go. Thank you.
Hello, everyone. Thank you, Oliver and Michael, and thank you to give me the opportunity to talk about our most recent data on the link between inflammation in COVID-19 patients and the activation of the complement pathway with a focus on the C5a C5aR1 axis. Here are my disclosures. I'm an employee of Innate Pharma. Just a few words before starting on the coronavirus disease 2019, also called COVID-19. This disease, as you know, appeared in China in December last year, with clusters of people with pneumonia in the city of Wuhan. The researchers in China did the sequencing of the virus, and they actually discovered that this virus was actually a new virus, and they called it severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2. As you know, this virus has affected millions of people worldwide, including more than 1 million deaths, and the COVID-19 disease was declared a pandemic by the World Health Organization on March early this year. Regarding the physiopathology of the disease, we know that most of the COVID-19 patients present only a few mild symptoms, with most of the patients that do not have any symptoms at all. But there is also 15% of the patients that progress to severe pneumonia and about 5% that develop acute respiratory distress syndrome, also called ARDS. What we know now is that the immune response plays a role in by contributing to the virus elimination, but also contributes to the development of ARDS. And there was, since the beginning of the pandemic, a tremendous effort by the scientific community to understand the immune response in COVID-19 patients. But there is still a need to decipher this response and identify the potential new targets leading to the excessive inflammatory response in some patients. So in Marseille, what we did is that we actually initiated a task force, including MDs and nurses from hospital in Marseille, scientists from academia including ex-Marseille University, the CIML, the Centre d'Immunologie de Marseille-Luminy, and Marseille Immunopol, and scientists from Innate Pharma. So it was basically in the middle of the lockdown in France, and it was a group of 40 to 50 people that worked basically 24/7 to generate a lot of data that led to the recent publication in Nature of this paper named Association of COVID-19 Inflammation with Activation of C5a C5aR1 axis. And today I'm going to talk and present the data that we published recently. So thanks to our collaboration with hospital in Marseille, as I said, we had access to patient samples. Blood first of 82 patients, healthy controls, and COVID-19 patients, polysymptomatic patients, patients with pneumonia, and patients with ARDS. For the COVID-19 patient, we had access to 3 time points, T0, which is basically the date of the diagnosis, T1 and T2, that is approximately day 5 and day 10 after the diagnosis. From the blood, we had access to the sera, and we perform ELISA, and we had access to the PBMCs, and we perform flow cytometry analysis and functional assays. We had also access to lung samples, lung biopsies, and we perform multiplex and chromogenic IHC, and on the bronchoalveolar lavage fluids, or BALFs, we perform flow cytometry and ELISA. What we saw first actually is that the disease severity was actually associated with an increase of the inflammation in patients. As you can see on the left side on this slide, with the CRP level, the C-reactive protein level in the sera, you can see that patients with ARDS and patients with pneumonia have very strong CRP level compared with patients with very few mild symptoms. Then we looked at the pro-inflammatory cytokines and chemokines, the usual suspects. As you can see here, there is, in patients with ARDS, high level of IL-6, CCL4, CCL2, and CXL9. But what you can also saw in these graphs is that for IL-6, particularly, there are several patients with ARDS with low level of IL-6 in the blood, meaning 2 things. The first one is that maybe that IL-6 is not a good marker for the disease severity, and the second thing is that maybe IL-6 is not the best target for the COVID-19 patients. At Innate Pharma, we have a very strong interest in the complement pathway, and particularly in the C5a C5aR1 axis. Just few words about the complement pathway. You know that there is the classical, the lectin, and the alternative pathway in the complement, and they all lead to the cleavage of C3 and the cleavage of C5. The cleavage of C5 is leading to the formation of C5b that will bind C6, C7, C8, and C9, leading to the formation of the membrane attack complex, which is a very important mechanism to eliminate bacteria. The cleavage of C5 is also leading to the production of C5a, that is a very important molecule, an anaphylatoxin actually, with inflammatory and properties, and also involved in the coagulation. And what we saw by looking at the C5a level in the blood of healthy controls and COVID-19 patients is that there is a very strong association between the disease severity and the C5a level in the blood. And the all the patients with ARDS actually have a very strong C5a level in the blood. What we saw also, and this is what we saw on the graph on the right, is that the level of C5a was quite stable throughout the course of the disease. And what we know now is actually the SARS-CoV-2, the virus itself, can activate directly the complement pathway. And there are many papers now describing this phenomenon. The virus can activate the complement pathway, leading to the cleavage of C3, the cleavage of C5, and the release of C5a that will bind its main receptor, the C5aR1, but also the C5aR2, which is less expressed and with a different role in the complement pathway. But at that time, when we started our work, the consequences of the activation of the complement pathway in COVID-19 patients was actually unknown. So what we looked then was the expression of the receptor on immune cells, and especially on neutrophils and monocytes. And as you can see here, C5aR1, the main receptor of the C5a, is actually quite strongly expressed on the neutrophil and the monocytes in healthy controls and in patients with COVID-19. Regarding the biological effect of the C5a, what we saw, and it was actually a very simple test, is that we took monocytes from COVID-19 patients and activate them overnight with LPS or R848 or resiquimod, which is a TLR7/8 agonist. We saw that we were able, and this is known, to induce production of IL-6, TNF, and CCL2 by the monocytes. And when we added C5a, the production of those pro-inflammatory cytokines was much stronger by the monocytes, meaning that C5a is actually a very potent molecule to induce pro-inflammatory cytokines production. Then we look at the lung, because we saw that the C5a is present in the blood and the C5aR1 cells are present also in the blood. But what about the lung, which is one of the first and main target of the disease? So we reanalyzed RNA-seq data from published study early this year. As you can see on the UMAP on the left, is that the valves from healthy controls and the valves from ARDS, they cluster in a different way. And when we looked at the RNA level of pro-inflammatory cytokines, we saw that there is actually a strong difference between the RNA level in the valve from healthy controls and in the valve from ARDS, with much more expression in the valve from ARDS, obviously. Regarding the expression of C5aR1, we saw that actually this RNA was found in both healthy controls and ARDS patients, meaning that in valve from ARDS, we are able to find C5aR1 positive inflammatory cells. So this was at the RNA level, and we looked now at the protein level in our cohort of patients. And what we saw, and actually we were able to repeat what we saw with the RNA, is that C5aR1 was strongly expressed on neutrophils and monocytes in the valves of COVID-19 patients. And then we looked at the level of several pro-inflammatory cytokines and chemokines, and what we saw is that in the valves from COVID-19 patients, there is a quite strong expression and level of C5a, but also CXCL9, CCL2, and CXCL8. Regarding IL-6, as you can see on this graph, the level of IL-6 in the valves were not that high, actually. And thanks to our collaboration with hospital in Marseille, we had access to lung sample from COVID-19 negative and COVID-19 positive patients. What you can see on this slide is actually a macroscopic view of a lung from COVID-19 and COVID negative and COVID-19 positive patients. And on the right side, you can see this is what you saw usually with the allograft rejection of a lung And this is what we observed also in COVID-19 patients. Now, using the multiplex LC technology developed by Aquarius, we looked within the tissue, within the lung, the organization of the cells that were expressing C5aR. So on the left, you can see an example of COVID-19 negative patients with very few macrophages, CD68 in red, and some of them express CD163, a typical M2 marker, and few of them express C5aR in green. Now, when we looked at the COVID-19 patients, you can see that the image is completely different. This is a different situation with a lot of macrophages, CD68. A lot of them are expressing CD163 and also C5aR. We then quantified the number of cells that were present on the section using Halo, and what we saw, and this is on COVID-19 negative patients first, is that very few C5aR were actually present in the lung, and the majority of macrophages were actually C5aR1 negative. Now, looking at the COVID-19 positive patient, what we saw is that the number of CD68 was actually quite strong, and the majority of cells that we found were triple positive, CD68, CD163, and C5aR. And there was actually 10 times more C5aR positive in the lung from COVID-19 patients compared to COVID-19 negative patients. And what we also saw is that by looking at the arteries in the lung from COVID-19 negative and COVID-19 positive patients, is that in some patients, in some COVID-19 patients, we saw what is called obliterating endarteritis lesion, meaning that you have an artery with a thrombus in the middle. This is what you saw on this slide, actually. And these arteries that are clogged with the thrombus are actually full of macrophages, and some of them express C5aR. And close to the clogged artery, you have also macrophages that are packed, islets of macrophages and most of them express C5aR. And what we also saw is that in those obliterating endarteritis lesions, there is an activation of the complement pathway, as you can see here, with the deposition of the C5b9 on the wall of the arteries. So in conclusion, what we saw so far is that in most of the patients that have a non-severe COVID-19 disease, that the virus is entering within the lung, is binding ACE2 receptor on the pneumocytes, and this leads to the production of C5a. C5a is going to bind its receptor expressed on neutrophil and monocytes and will induce the production of pro-inflammatory cytokines. For some patients, the ones that have a severe COVID-19 disease, the virus is entering the same way in the lung, but there is an overproduction of C5a that will recruit monocytes and macrophages and induce a strong production of pro-inflammatory cytokines. Within the capillaries, there is also actually a strong immune response leading to the formation of thrombus. And what we wanted to know is that, is there a way to come back to a more normal situation in patients with severe COVID-19? So we had the idea to repurpose one of our monoclonal antibody used in the clinic in oncology that is blocking the C5aR1. This antibody is called avdoralimab, and we looked at its effect on the activation of myeloid cells. So here what you saw is the baseline level of CD11b on the myeloid cells. When we added human C5a on top of the myeloid cells, you can see that there is actually an increase in the expression of CD11b, meaning that we were able to activate the myeloid cells with human C5a. And by adding avdoralimab, the blocking antibody, which is a Fc silent monoclonal antibody, we were able to come back to a normal CD11B expression level. Here, this is basically the same data, but we increase the concentration of C5a, and you can see that the more C5a you put, the stronger expression of C5b we obtain. Here is when we used avduralumab, and you can see that we were able to control the CD11b expression by adding avduralumab. Regarding the migration of the cells, we actually looked at neutrophils and we used a simple assay with a transwell assay. And what we saw is that by putting human C5a, we were able to recruit neutrophils. And when we added avduralumab, our monoclonal antibody, we were able to block the recruitment of and the migration of neutrophils. And this effect was lost actually when we used the isotype control. So in conclusion, what we saw actually is that the targeting of C5a can actually block the myeloid cell activation and the migration of the inflammatory immune cells. We then decided to look in vivo using a human C5aR1 knock-in mouse model. And we first controlled the replacement of the wild-type mouse C5aR1 by human C5aR1. And as you can see here is that in this knock-in mouse, you have expression of human C5aR1 on CD11B, as it should be, and also on monocytes and neutrophils, as you can see on the bottom panel, and with a similar expression to that observed with the mouse C5aR1. We took this model and we developed a C5a-induced acute lung injury or ALI model by adding C5a in the system. So with the mice, here the mouse in white, we perform intranasal instillation of PBS. And as you can see on the histology image, there is actually a normal lung with normal alveoli in this model. Instead of PBS, if we perform the instillation with the human C5a, this is the mouse in gray, you can see that there is actually quite strong inflammation in the lung with the recruitment of inflammatory immune cells and the presence of blood within the lung. And then if we treated the mouse with avduralumab prior instillation with human C5a, you can see that you can control the immune response and the lung come back to normal with normal alveoli, and you can reduce the amount of inflammatory immune cells within the lung. And if you used instead of avduralumab isotype control, you can see that the effect is lost actually, and there is still a very strong inflammation within the lung of those human C5aR1 knock-in mouse. And by analyzing the BALFs of those mice, what we saw is basically exactly the same, is that when we perform the human C5a intranasal instillation, we were able to induce the recruitment of CD45 positive cells, immune cells within the lung, and particularly the LY6G monocytes and LY6G negative neutrophils within the lung. By adding avduralumab, we were able actually to come back to a normal situation, similar to that found with instillation of this PBS. And this effect with avduralumab was actually lost when we used only an isotype control. Regarding the albumin level, which is a good marker of the lung damage, we are able to find that avduralumab was able to control and to decrease the albumin level, meaning that there is much less lung damage in the mice treated with avduralumab. Let's come back to patients. What we saw is that when we used monocyte from COVID-19 patients activated overnight with R848, TLR7/8 agonist, If you remember, by adding AR848, we were able to induce the production of IL-6, TNF, and CCL2 by the monocytes. By adding C5a, we were able to increase even more this production. But then when we add avduralumab, our blocking antibody, we were able to decrease this level with our blocking antibody compared to that observed with isotype control. Meaning that avduralumab is working to control the lung inflammation in mouse, but also on human cells here on monocytes. To summarize what we hope is going to happen in patients treated with avduralumab, if they are not treated, currently what they encounter is that there is a strong pro-inflammatory cytokine production within the lung, with the formation of thrombus within the capillaries and the arteries. But by treating them with avduralumab, we think that we can control the recruitment and decrease the recruitment of pro-inflammatory cells such as neutrophils, monocytes, and macrophages, and decrease the production of inflammatory cytokines. In the capillaries, we can control the formation of thrombus because of the control of the production of inflammatory cytokines and the recruitment of pro-inflammatory cells within the capillaries and arteries. It was in the lung, but now we looked also at other organs, because you know that the immune response is not controlled, is out of control in the lung, obviously, but also in other organs. So we had access to liver samples, but also kidney and other organs from COVID-19 patients that actually died from the disease. And what we saw using the multiplex LC technology developed by Aquarius Biosciences is what we were able to find a lot of macrophages within the liver using the CD68 marker. Those macrophages also express CD163, but they also express C5aR at high level. So this means that using avduralumab, our blocking antibody, we can control the overinflammation within the lung, but also in other organs that are affected by this disease. So I will finish this presentation with a clinical trial we started several months ago called FORCE for COVID-19 elimination, which is the double-blind randomized study versus placebo of the avduralumab in patients with severe COVID-19 disease. This is a study design. So we have two cohorts. Cohort 1 with 60 patients, comprising COVID-19 patients with severe pneumonia who don't require hospitalization in ICU. Those patients with randomly received avduralumab plus best supportive care or placebo plus best supportive care. The second cohort of around 50 patients will be comprising of COVID-19 related ARDS patients hospitalized in ICU, and they will receive either avduralumab plus best supportive care or placebo plus best supportive care. And the primary endpoints are indicated in this slide. Finally, I would like to thank all the patients and their families, people from Innate Pharma, and especially Eric Vivier that led this work, and Olivier de Maria, Julien Carvalli from hospital in Marseille, Frédéric Velli from Marseille Immunopol, and also people from my team, Laura Sant-Miranda and Flavien Caragel, that did all the multiplex IHC analysis and quantification. And I will take any question that you might have. Thank you for your attention.
Thank you very much for that insight into the Explore COVID-19 study and into the role of the C5a C5aR1 axis. Okay, our first question is for Romain. how quick was the multiplexed IHC panel development?
So actually, as I said, this is a good question because the Explore COVID, as I said, the project lasted a month and a half or so, and it was too short to develop a new multiplex IHC panel for this specific study. And also it was in the middle of the lockdown in France, and it was quite complicated, not impossible, but complicated to order new reagents. So we decided, as we repurposed the avdoralimab that is currently tested in oncology, we also repurposed the multiplex IHC panels that were developed for the ongoing clinical trial of avdoralimab plus durvalumab in HCC and non-small cell lung cancer. So we had to do some minor changes in the protocol, and we were ready to apply the multiplex IHC protocols on the organ from COVID-19 patients we received, in the lungs, the liver, and I show you the data, the kidney, the heart, and the brain as well. And we did that in less than a week, which was very quick. And if you are familiar with the multiplex IHC development, you know that it's very quick. But generally speaking, we usually develop from scratch 7-plex IHC panel in around 9 weeks, I would say, usually, which is, I think, optimal, and we cannot reduce this time for the development.
Okay, great. Thank you very much. Second question is to Oliver. Do you have antibodies to detect COVID-19?
Thank you for that question. We are developing 3 antibodies right now to detect the spike protein, the nucleocapsid protein, and ACE2 receptor in the tissue to which these molecules, especially the spike, should bind. These antibodies are being developed for our Codex platform, and they're expected to come out early next year. With regards to Phenomics, we are seeing reports now or publications that are reporting certain antibody clones against spike protein. So, for example, just a few weeks ago, there was a paper by the lab of Ramin Saleh Rad and Jocelyn Kim at the David Geffen School of Medicine at UCLA that has shown very nice staining with spike protein as well as an ACE2 receptor, which allowed them to look at lung tissue and co-localize both immune cell markers plus the pathogen or the COVID-19 virus. So it's in development very much, and more antibodies are becoming available as I'm speaking right now. Thank you for that question.
Excellent, thank you. Another one for Romain. What is the C5aR expression profile in healthy patients?
So thank you for this question. So in healthy patients, C5aR1 is highly expressed, as you saw, on neutrophils and also on non-classical monocytes. It's also expressed on basophils at intermediate level and also on classical monocytes. It is also found on eosinophils, but at lower level. And what we know also, because C5aR1 is a molecule of interest for Innate Pharma, is that it is not expressed on B cells or T cells or dendritic cells, for example. But what we also know is that it's also expressed on few normal non-immune cells, and especially it's found in normal tubular epithelial cells in the kidney, for example. And in COVID patients, as you saw, there is a similar expression for C5aR1 with a strong expression on neutrophils and monocytes and macrophages as well in the tissues, and it is not expressed on lymphocytes, on T cells. This means by blocking the C5aR1, actually, we should be able to block the recruitment of inflammatory immune cells that lead to the ARDS development in some patients, but we are not going to limit the infiltration of lymphocytes, and especially of T cells that are important in controlling the disease.
Okay, thank you very much. another one for you, Romain. How did you quantify the cell densities in your IHC experiment in healthy versus diseased lung?
Yes, so this is a good question. So maybe I can describe rapidly the process we have for the staining. So basically, all our multiplex IHC staining are performed on the Leica-bound RX stainer, and the slides are scanned with the Vectrapol RX we have. Then we do the spectral deconvolution on Inform, and we do the fusion, the stitching of the mosaic images that are generated by Inform using in-house developed software we have at Innate. And then for the analysis, so with this in-house developed software, we can generate TIFF image format, and we can analyze them using Halo. And for the multiplex IHC, we usually use a high-plex module on Halo, and we then can export the data, the CSV file, and then we can process the data using our software.
Perfect, thank you. For Oliver, what kinds of tissues can be studied with Aquarius imaging technology?
Thank you for the question. The tissues that you can study with Aquarius technology are essentially more or less the same that you would be able to study with any other epifluorescence imaging technology, given that what we do is based on fluorescence imaging. So with Codex, for example, you can image fresh frozen or FFPE human or mouse tissue. We've tried many different organs, ranging from brain to spleen to lungs to skin even, and all of these perform quite well. So it's really just whatever you can image with a microscope. Phenomics platform is very commonly used in lung and various types of cancer tissues, and it's generally used with FFPE and human tissues. So it's very, very common in pathological samples, clinical pathology, because it allows you to do whole slide scanning. So you can take big pieces of tissue and you can scan them very rapidly. So there seems to be no real limit as to what the tissue is, as long as it's compatible with conventional histology protocols and conventional imaging hardware, such as carrier slides and optical so being sectioned. So we have a limit on how thick tissues can be. They need to be sectioned, but other than that, it seems to be very, very widely applicable. Thank you.
Okay, that makes sense. Thank you very much. One last one for Romain. Could you please give us an update on your clinical trial?
Yeah, thank you for this question. Actually, I wish I could, but actually, as you know, this is a double-blind clinical trial, and we are still blind for now. And I think this is a good way to perform a clinical trial. And as you can imagine, at the beginning of this clinical trial, we had some issues when we first started, because there was too few patients hospitalized after the lockdown in France, which was good. And now, as you all know, we are now facing a second wave in France and more generally in Europe. So obviously, we are now recruiting more and more patients, and it's becoming very quick now. So I cannot give you a precise date for the data, but stay tuned. We should be able to communicate on the data of this clinical trial very soon, I hope.
Okay, great. Thank you very much. I'm afraid we're going to have to leave it there, but again, let me reassure you, any questions left unanswered today will be answered after the event by email. The webinar will also be available on demand in just a few days' time, so you'll receive an email alert when that's ready. For now, thank you once again to Oliver and to Romain for presenting, and thanks to everyone for joining us today. I really look forward to seeing you at future webinars, so goodbye.